Uh, Cindy, okay, you are recording. Okay, great. Good afternoon, everyone, and good evening, Professor Fanning. It's my great pleasure and honor, actually, to have Professor Fanning to give us a talk today in our department research seminar series at IIT in Chicago. I actually invited a few colleagues of mine from the FDA and from other academic institutes. So you probably see names popping up from everywhere, from University of Georgia, from University of Maryland, and from University of British Columbia as well in Canada. Let me just read through the bio for Professor Fanning. So Professor Fanning graduated from University College Cork with a bachelor's degree in biochemistry and a PhD degree in molecular microbiology. And Professor Fanning is currently the director for the UCD Center for Food Safety. His research interest expertise is in the antimicrobial resistance, bacterial transmission across the food chain, and with a focus on understanding the mechanisms of transmission, adaptation of bacteria to food production environments, using different molecular approaches, including the next generation sequencing methods. Professor Fanning's research group is very well known because I've reading so many papers from your group in Salmonella and in particular Chronobacter species such as Sakizakii, in low moisture foods such as powdered infant formula. Professor Fanning has published more than 300 research papers, 32 review articles, 28 book chapters, and 2 books. Wow, that's just too impressive. I don't know how you manage your time. He currently serves on the editorial boards for many international journals. Many of us are very familiar with the Foodborne Pathogen Disease, Journal of Food Protection, and so on. And also, he is a fellow of the American Academy of Microbiology, which is a very prestigious title to recognize an outstanding world leader in microbiology. So with that, I would like to leave to Professor Fanning to start the talk. We'll save the question session at the end of the seminar. So you can either unmute yourself or you can text in the text box, in the chat box. And with that, Professor Fanning, please.
So I hope everyone can see this.
Yes.
See it? Okay, great. Well, thank you, Dr. Zhang, for a very kind introduction. Far more flattering than I deserve, I have to say. And looking at the audience that are listening to us, there are far better experts on the line than I. So I'm honored to give you this presentation this evening. And when you asked me to do this, I thought it might be interesting just to take a small perspective look at our work, and not just our work, but also the work of Ben Tall, who's on the line, and Gopal Gopinatharo, who's also on the line from FDA, and others that we've worked with over the last number of years. And it's been an absolute pleasure working with them. So thank you again for this kind invitation. I'm really delighted to be with you this evening. It's actually very dark outside, so our evening, and I guess your morning. So I thought I'd say something about the use of next generation sequencing methods to study surveillance. And I'm probably not going to say too much about surveillance, but more or less to look at the study of bacterial adaptation. And it's really just a kind of a retrospective look at what we've been doing in UCD since about 2005, when we started working in this area. So just to give you a little bit of background in an Irish context, and this is very much an Irish focused, if you like, effort. We're a very small country, about 5 million people or so, but yet we seem to be one of the leading world producers of infant formula. So about reckon about 10% of the world's powdered infant formula comes from Ireland. And I guess that's not really surprising when you think about the nature of our farming activities on the island. I mean, it's very much a grass-fed approach to our cattle and growing our cattle. And as you can see here from some of the metrics that I have in my slide, there's quite a lot of money tied up in infant nutritionals. In fact, if you think about the agri-food sector in Ireland, it's probably at the peak of the sector in terms of its value towards the Irish economy. So it's one that the government want protected, and they want to make sure that there's no issues associated with this particular important industry. And given the fact that the nature of the consumer here is a very vulnerable consumer, both at the extremities of age, and I'll come back to that in a minute. But on the island, we have 3 production sites that are responsible for delivering this supply chain of powdered infant formula. So on the left-hand side of my slide, you can see Abbott, and I'm sure you're very familiar with Abbott. It's a US company, and I'll come back to them in a moment. We also have Danone, who are located in the south of the country, and Nestle, what used to be a Pfizer plant, located on the west coast of Ireland. So these 3 plants produce this amount of infant formula that I'm talking about. They take a substantial amount of the milk supply that's produced every year from the cattle that are grown on grass, as I've described. So moving on again, when we think about issues then that arise in terms of why we get adverse events associated with the powdered infant formula supply chain, it mainly has to do with contamination. And some time ago, Jeff Farber and colleagues produced this very nice figure, which I tend to use from time to time when I'm talking about this. And it's a really nice way of trying to get an overview of the potential risk factors, at least some of them, if not all of them, associated with this production chain. And broadly speaking, these can be divided into factors that are associated with what we call intrinsic contamination. So this is where the supply chain becomes contaminated during production. And then the other part of that is, of course, extrinsic contamination, where you get a contamination happening after the product has left the factory and has gone to the home or the hospital, where perhaps controls or hygiene measures are not appropriately adhered to. And so this gives rise to contaminated powder, which can then be fed to a vulnerable infant, and it can cause infection. But when we look again at the intrinsic contamination and perhaps tease out some more of the reasons underpinning that, you can see here in my slide on the left-hand side that I've listed a number of challenges that the bacterium, in this context, either Chronobacter or Salmonella, must overcome in order to survive within that very hostile environment. So there are things like heat. If you've ever been inside a powdered infant formula production site, it's a rather warm place. An ambient temperature of well above 30 degrees when the factory is running full production. It's also a low moisture environment, so there's not much scope for a bacterium to thrive in those kinds of conditions. There's limited nutrients, and the quality control people are always spraying sanitizers around the factory to try to make sure that they're killing any biological material around. And there's also a microbiome here, which presumably is also exerting some type of control. And we've seen a little bit of an insight into that over the last number of years. So these are just some of the routes of contamination that you can find with respect to the powdered infant formula production chain. And as I say, for Chronobacter to survive, it must overcome those, or better put, it must adapt to those conditions. So when we think about bacterial adaptation then, in a low moisture context, there are a couple of things we can say about it. For example, if we were to go out on the street in Chicago today and do a quick head count survey and ask people what they thought about the risk of infection associated with normal foods versus, let's say, the risk associated with what we might call low moisture foods, I suspect they might say that low moisture foods should present a lower risk. I'm not entirely sure that that's actually true, because in fact, low moisture foods are really not sterile. And we've begun to really appreciate that more so because we've seen several outbreaks associated with these types of foods. The second thing to say about this is that we really don't know very much about how bacteria behave under these conditions. And so we have to assume that the physiology of a bacterium changes quite substantially, and therefore the kind of countermeasures we have in place or the risk mitigation measures may not necessarily be sufficient to deal with that altered sort of phenotype. And that brings into question things like hygiene protocols that are perhaps developed to deal with planktonically growing bacteria in an environment versus those that may be behaving differently in a low moisture environment. And similarly, with respect to manufacturing practices, you'd have to question those as well. So in the context of powdered infant formula then, there's really two bacterial species that stand out head and shoulders above everything else. And that is Chronobacter species. And again, I'll come back to the Abbott situation in a moment, but also Salmonella species. And here in Europe, you've probably heard about the Kinder chocolate egg outbreak from the Ferrero company, which was associated with the detection of a monophasic Salmonella in these Kinder eggs that the young kids will consume, their chocolate eggs that young kids will consume. I suppose the common feature about these types of foods is the fact that they're low moisture foods coming from a built low moisture production environment. And again, this slide just illustrates those typical types of foods, the types of outbreaks that have been reported over the last number of years. For example, Salmonella in chocolate, in nuts is well known, Bacillus cereus in various kinds of foods, Clostridium associated with honey, and then Shiga toxin producing E. coli strains. And there's even some concerns around Listeria as to whether or not Listeria can actually behave in a similar manner. But all of these foods are what we call low moisture foods. And low moisture foods, as defined by our colleagues in the FDA, would be foods whereby the water activity is below 0.85. And I suppose, you know, for any of the students listening to us, it's a typical question that they might get asked in a viva: what bacterium can survive and grow at a low moisture activity of 0.85? Well, there's one that stands out, and that's Staphylococcus aureus. Everything else that we talk about, Salmonella, Shiga toxin producing E. coli's, Chronobacter, they require a much higher AW value in order to grow, but not necessarily to persist. And that's the distinction. And I suppose that's why we think about this as an adaptation type phenomenon. So just to sort of take a slight aside for a second, I just want to show you something completely left of field. So this is a figure which Mark Ackman, for those of you who know him, he used to run the EnteroBase database located at the University of Warwick in the United Kingdom. And he published a paper last year where he provided a genomic summary of the diversity of some more than 32,000 pre-2011 Salmonella enterica strains that were lodged in this Entero database. And I've put an arrow against Mbandaca, so Salmonella Mbandaca. And I've done that deliberately because when you pull out Salmonella Mbandaca from the EnteroBase repository and you construct a phylogenetic tree that hopefully looks something like this, there's two arms of this tree that I want to draw your attention to. There's this arm here on the right-hand side, sorry, on the left-hand side in dark blue, which are a series of isolates that we've dealt with in Ireland. And then, sorry, pardon me, there's another one here in lighter blue, which are also a series of isolates which come from the northern part of Ireland. So here you can see a kind of a geographical distribution of different kinds of Salmonella Mbandaca that have been reported through EnteroBase over the last number of years, as I say, pre-2011. The interesting thing about it is we have a situation at the moment where we have a production site that's colonized by Salmonella enterica serotype Mbandaca. And it speaks to the kind of adaptive nature that you see in some of these bacteria. And in this particular context, what's very unusual and that has happened is that the Salmonella Mbandaca strains that are colonizing this particular production site have acquired a completely intact lactose operon. So again, a question for students in Aviva: what's the lactose phenotype of Salmonella? Of course, it should be lac negative. They're not supposed to be able to use lactose as a carbon source. But the adaptation in this particular case to a production site that's involved in dairying has necessitated the acquisition of a lactose operon encoded on an ICE element, a complete ICE element, which is an intergenic massive piece of DNA, which contains phage elements and also transfer elements. And that has become integrated, as you can see here in the middle part of my slide, right into the genome of Salmonella enterica and Bendaca. And that particular ICE element has been acquired from a Leclercia bacterium. So the whole idea of adaptation is something perhaps that we need to think a little bit more about and try to tease out the reasons as to why this happens. And I suppose that's what brings us then to the Abbott situation at the moment and the topic for our conversation this evening, which is about Chronobacter and powdered infant formula, and the question as to why we see this organism adapting to these kinds of low moisture environments. Well, I don't think I need to rehearse the scenario at the moment with respect to this particular outbreak. I think it's well known in the US and outside the US. Unfortunately, 2 of the 4 infants that were diagnosed with this infection have died. And this is one of the absolutely sad pieces associated with this particular type of food chain. And it shines the spotlight in a big way on the manufacturer in terms of what could have gone wrong and how do you mitigate this to prevent any future occurrences. So I suppose the scenario we're describing here is one whereby a young infant consumes a bottle, a made-up bottle, containing a high dose of this particular bacterium, Chronobacter. And I'm going to focus just on Chronobacter for the rest of my presentation. But the infant we're describing doesn't look anything like this infant in the picture that I'm showing you, because we're talking about a neonate which really is much, much weaker looking and much more vulnerable than the baby that's in my picture here. But just for the point of view of illustration, you can see here the kinds of events, I suppose, biologically that happen when an infant consumes a high dose of a lethal pathogen. So the bacterium would normally go down through the alimentary canal, like all food. It probably can escape the acidity of the stomach, because in a young infant, the acidity of the stomach is probably at around 5. So that's not really presenting much of a challenge. And then the bacterium moves on down lower into the intestinal tract, attaching to the intestinal epithelium, where there may be some leakiness in the barrier at the epithelium, giving rise to a necrotic enterocolitis. Then the organism may cross the barrier, getting into the vasculature system, where it may cause a bacteremia or a sepsis. It's translocated around the body until eventually it presents itself at the blood-brain barrier. And this is where then certain strains of Chronobacter have been shown to have the capacity to cross that blood-brain barrier, mainly mediated by the expression of this outer membrane protein or outer membrane porin called OMP-A, crosses the barrier and then causes an infection in the meninges of the baby. And in nearly all of those cases, they're fatal. So trying to understand the nature of the adaptation in the factory and then what happens in the baby is a very big challenge to both the producers of powdered infant formula and to us as scientists that are interested in investigating this. So taking a historical look at this, Anna Bowen in CDC in about 2006 published this paper where she took a retrospective look at the cases that were on file in CDC at the time, with the view to trying to see, could she establish any evidence to show that there was a link between some of these Enterobacter sakazaki infections, as they were called at that time, and the evidence of a contaminated powdered infant formula being fed to those babies. So in this particular instance, Anna identified about 46 cases that could be stratified into those that were presenting with meningitis. And you can see the typical presentation of those infants. They were at near-term normal birth weight for birth at about 2.5 kilograms, almost a full gestational period, and infection developing after about 6 days. On the other hand, there were bacteremic cases where the babies were very much of low birth weight, so not really full gestational period, and infection then developing after about 35 days. So on my slide here on the top right-hand side, you can really see the type of baby that I'm referring to, a very vulnerable infant, as I said earlier on, tube fed, lots of different life support systems here going on. So really a very debilitated infant with a very compromised immune system. So it's not surprising that when you present a high dose of a bacterium that you get the type of devastating response as a result. In many of these cases, Anna was able to show that there was a link between the clinical presentation, in other words, the culturing of the organism from the cerebrospinal fluid or the feces, and then the matching of that organism with respect to culturing of the same bug from the contaminated batch of powdered infant formula. And that's in a sense an ideal epidemiological story that appears to be reasonably watertight. So insofar as we're currently situated then, there are some characteristics of the foodborne illness that we can say we know something about. So for example, I've mentioned some of the symptoms: necrotizing enterocolitis, I've mentioned bacteremia and meningitis. For those infants that recover from these infections, there's usually some type of neurological damage, such as delayed development or hydrocephalus. But there's still a lot about this bacterium we don't know about. For example, the infectious dose, we're not entirely sure how much of this organism you need to consume in order to become fatally infected. And from an epidemiological point of view, the food that's most often implicated is infant milk formula. And in fact, I could have added a second heading here, which is the food most frequently implicated for older people, because there is a risk factor there as well, as you'll see from this slide. And again, this was a follow-up study, again from CDC, published in 2014 in Emerging Infectious Diseases, which clearly showed a second group of individuals that we also needed to pay attention to. And those are folks like myself at the extremities of the age on the right-hand side here, who might have a compromised swallowing mechanism and who might therefore need to have medicated foods provided. And of course, infant formula is, if you like, a medicated food. Similarly, for older people, they take medicated foods. The same risk factors present to both. However, the illness is much more severe in the case of the infants, perhaps less so in the case of the older individual. And so perhaps to some extent, that's why heretofore it may have not been readily recognized as presenting a problem to older people at the extremities of age. It also begs the question as to where this bacterium comes from. And we still don't really know the answer to that. Nevertheless, our colleagues, Professor Roger Stephan and his team at the University of Zurich, some years ago presented this paper, or published this paper, I should say, in Research Microbiology at the Pasteur Institute. And they very elegantly showed that, in fact, if you looked at the bacterium and you could stain it, you could see that when you co-cultured it with certain types of plants, it endophytosed into the plant such that it became part and parcel of the plant structure. So you can see this here from the confocal microscopy in panel D of my figure. So it appears that Chronobacter can endophytically colonize the roots of tomato and maize plants. And when you think about it, there's probably a lot of truth in that, because some of the attributes of the bacterium, such as its yellow pigmentation, as it's been euphemistically known for, may be a kind of a defense mechanism that the organism expresses when it's out in the environment. But to some extent, epidemiologically, this kind of fits with the broad picture as to where we expect to find high numbers of this organism, particularly in association with plant products, and more so dried plant products such as dried herbs. So just to kind of give you a quick snapshot then of what I'll talk about some of the biology and the background to Chronobacter species, and then I'll mention something about the adaptation to the production environment and how we then started to try to tease apart what the bacterium was doing once it entered these low moisture conditions, and then I'll try to end up with some conclusions. So just to take a look quickly at the biology, well, I think as everybody knows, the organism is a member of the Enterobacteriaceae family, therefore it's a gram-negative rod-shaped bacterium that's facultatively anaerobic. And up until about the mid-2000s, the taxonomy of this bacterium was in a state of flux, if I could describe it like that, being originally described as Enterobacter sakazaki, named after the Japanese microbiologist Rikiki Sakazaki, who described this organism prior to 1980. Since that time, it has now been, the taxonomy has now been revised, and we have a new genus called Chronobacter species that currently consists of 7 subspecies, although there will be, I expect, reports soon of some new variants within that 7 subspecies category. From a laboratory point of view, the organism is very easy to deal with. However, if you were to say, well, what is one of its most important attributes? I think the next bullet point describes that. And in my opinion, one of its most important attributes is its zero tolerance. In other words, its ability to withstand desiccation. And that fits again with the epidemiological picture that we see, the association of this bacterium with a low moisture type food, such as powdered infant formula, where the organism is not necessarily growing because there's really no moisture there, but it is able to persist. And when water is introduced to that powdered infant formula, you get the scenario that I tried to describe earlier on with respect to the baby consuming that bottle of made formula. And so this gives rise to rare opportunistic infections that perhaps up to a number of years ago, clinical microbiologists may not have recognized, the symptoms I've mentioned before, and then the link with urinary tract infections with older people and wound infections in older people is also something I've briefly touched on. So this is what it looks like. On the left-hand side, you see that yellow pigmentation. And so historically, Enterobacter sakazaki would have been referred to as a yellow pigmented Enterobacter cloacae, because I suppose when you don't understand the taxonomy of an organism you've isolated, you call it Enterobacter something or other, and it just sits in that particular genus until such time as somebody goes and takes a more detailed look at it. On the right-hand side of my slide here, you can see a very nicely flagellar stained version of the organism. And as I said in the previous slide, it is a motile bacterium. So I'm tending to bang on a little bit about the adaptation, and that really is important because we need to understand how Chronobacter species become adapted to different environmental niches. So if we take this very nice figure from a paper that was published in a review earlier this year, there's probably 3 key stages related to the adaptive mechanisms. And I'll touch on this again briefly as I go through the pursuing slides. So if we think about environmental persistence then, and we say, well, what kind of attributes do we think a bacterium should express in order for it to enable the survival within that environment? Well, things like biofilm formation could be important because we know that in biofilms, all bets are off in terms of sanitizing agents. Adaptation to high temperatures, that could be a good one, particularly in light of what I said about the powdered infant formula production environment. Tolerance to acidic conditions could be good. Osmotic tolerance, excellent. So osmotic tolerance would really be important, particularly when you're talking about a low moisture environment. And then if you move on counterclockwise through this, you come to the infection strategy. And then again, what attributes do you need here? Things like motility, perhaps quorum sensing, adhesion, colonization, and invasion, translocation would all seem to be important things for this bacterium to cross the intestinal barrier and then eventually to cross the blood-brain barrier. And then, of course, what about systemic persistence? Well, your ability to invade or evade the immune system, and particularly your ability to survive in macrophages. So all of those attributes are called into play depending on where this organism is in an environmental niche. And all of those attributes are underpinned by genes of one kind or another that become expressed and then become switched off. And so it's trying to understand how that process happens becomes relevant to trying to get a better picture as to how Chronobacter species behaves under these different conditions. We already know from various reviews that have been published over the last 20 years or so that we can find Chronobacter in a range of different kinds of foods right around the world and in different kinds of environments as well, mainly associated with the production of food. And in fact, our colleague and good friend on the line, Ben Tall, would have been one of the first teams to identify Chronobacter species in what Ben called filth flies at the time. And again, they were able to show recovery of Chronobacter from these kinds of vectors that would be transmitting the contamination around an environment. So in terms of environmental persistence, then, these are just some of the genes illustrated here schematically on the left-hand side that underpin some of those mechanisms. The genomic island that you see up here on the top right has to do with heat tolerance, biofilm encoding genes, various types of transcriptional factors. These potassium transporter functions become very important in conditions of low moisture, which I'll come back to later on. And then, of course, the importance of osmoprotectants such as trehalose, glycine, and proline are hugely important in terms of allowing the organism to survive when moisture is withdrawn from its ecological niche, as would be the case in a low moisture condition. Similarly, as I said, with respect to the infection in the host, the kinds of strategies here, the outer membrane proteins or porins, I've mentioned en bay in terms of the ability to cross the blood-brain barrier, and various other virulence mechanisms that are associated, such as the ability to withstand reactive oxygen and the use of the activity of the SOD enzyme or superoxide dismutase enzyme to negate the effects of radical species that are produced. So all of these are necessary functions in order to allow the bacterium to adapt to these ecological niches. They're driven by selection and they're underpinned by genetics. So when we step back then and we take a look at the taxonomy of Enterobacter sakazaki as it was, at the time, Dr. Carl Iverson, who worked with Professor Stefan in Zurich and then subsequently with us in Dublin, was able to show that if you actually amplified the 16S ribosomal RNA gene from these bacteria and you sequenced it and you compared the sequences, then you got a series of clusters that we hadn't recognized heretofore. And these clusters of isolates, Carol called at the time Group 1, Group 2, 3, and 4. And when this particular piece of work was done, we were able to show, or Carol was able to show, that Chronobacter sakazaki and Chronobacter melanoticus, as they became known, fitted nicely into Group 1. So it's kind of difficult to try to, if you like, separate those two. Then in Group 2, you had Chronobacter turicensis and Chronobacter universalis. In Group 3, you had Chronobacter mutagensii, which was named after a Dutch clinical microbiologist working at Nijmegen. And then, of course, because we were involved in the act, we had to get our name on the list as well. So we called the fourth one Chronobacter dubliniensis. And then, as I'll show you in the next slide in a moment, Ben Tall and his team very elegantly showed that, in fact, the most important species here are those associated with Groups 1 and Group 2. Groups 3 and Group 4 were mainly environmental contaminants that may not necessarily be big players in the clinical picture. But it was also important to sort of look at other organisms or other bacteria that were sharing the same ecological niches as to where we would find Enterobacter sakazaki, and to be able to distinguish between what was Enterobacter sakazaki or Chronobacter species versus everything else. So other bacteria at the time, which were also alpha-glucosidase positive, just like Chronobacter species, could be detected in these environments. And at the bottom of my slide here, you can see three of them: Cicibacter turicensis, which was originally known as Enterobacter turicensis, and then Franconibacter helveticus and pulveris, which was formerly known as Enterobacter helveticus and pulveris. You could see here that these were perhaps distantly related to Chronobacter species as such. They were alpha-glucosidase positive, but they could clearly be distinguished from those groupings, from the Chronobacter grouping, as I say, based on some of their genetic features. So why do we pick a name like Chronobacter? Well, you have to go back to Greek mythology for the answer to that. And this is mainly due to Carl Iverson's love of Greek mythology, I suppose. But in Greek mythology, you have a god called Kronos. And Kronos was the leader and the youngest of the Titans. And when Kronos overthrew his father Uranus, he ruled in a mythological golden age. And in order to protect his empire, he swallowed each of his children as soon as they were born. So I suppose the link here is between the death of those children as a result of Kronos eating them and deaths associated with infection linked to powdered infant formula, and hence the term or the genus name Chronobacter was born. And again, going back to Ben Tall's group at FDA, Ben and Gopal and the team there were able to elegantly show that if you constructed a SNP tree based on a very large collection of different Chronobacter species, as were at the time, you could show that on the basis of this SNP tree here versus the 16S ribosomal RNA tree that I showed you 2 slides back, the topology of the tree is largely conserved. So in fact, the 16S work at the very start predicted the differentiation of the different species within the genus. This was reinforced, as I say here, by the SNP tree from the FDA team, and also the distinction between those clinically important species such as Sakazaki, Melanoticus, and Turicensis versus the environmentally important species such as Mutagensii and Dubliniensis. And then these could clearly be differentiated from Franconibacter and Cicibacter, which I showed you earlier on. Again, there was some biochemical differences that you could use to make this distinction from the point of view of VIDAS, or those automated identification systems. And these, I think, and I'm sure, are inbuilt into that system at this stage. So it's possible to make these differentiations. So coming then to the nub of the problem, how does Chronobacter behave then in a food production environment? And can we understand what it is that underpins its survival within these types of niches? Well, the minimum AW value, again, which would be required to support the growth of Chronobacter, is estimated to be around 0.94. So that's very high. It's close to 1. Some Chronobacter species, as we've identified over the years, have become relatively resistant to heat. Now, that may or may not be related to that heat tolerance island that I mentioned a little while ago. Nevertheless, this heat tolerance dramatically increases when the bacterium is found in a low moisture condition. And in addition, this is also supported by the fat content in these foods. So typically those kinds of conditions that you might find in a powdered infant formula or in a dairy product. And not alone that, but Chronobacter is able to stay in this kind of a dormant or suspended condition, if you like, for weeks, months, and even years. So it's really, really an important challenge for the producers of powdered infant formula to recognize this fact and to do all in their power to make sure that their product and their environment is completely free of Chronobacter. So it begs the question then as to how does exposure of Chronobacter to these dry environments confer this adaptive phenotype? Well, one of the grad students in my lab some years ago, Chung Yan, took a look at some of the early bits of this. And what Chung did was she picked a number of different powdered infant formula production sites around Europe, and she surveilled them for a 26-month period. And during that time, she recovered as many of the Chronobacter species that were detected and characterized them, again, in collaboration with our colleagues at FDA and in Zurich. And what we were trying to do here was to genotypically and phenotypically characterize these isolates using a range of different tools at our disposal at the time. So molecular serotyping, various phenotype-based assays, pulsed field gel electrophoresis, which was really still used at the time when we did this study, multi-locus sequence typing. And then Ben and his colleagues had constructed this chrono array, which was particularly useful. So what Chung did here was she compared powdered infant formula associated isolates, which you can see here on the left-hand side of my graph, versus other clinical and control isolates, which we had in the collection in UCD and at FDA at the time. And again, this figure just provides an overall summary of this. And what it shows you here is the comparison of these isolates based on the different sequence types that we were able to identify, the pulsotypes we were able to identify, and then the differences we saw when these isolates were subjected to hybridization on this FDA chrono microarray. So what you see here, kind of going from left to right, is a major cluster down towards the middle, which is denoted by the sequence type 4. Now, sequence type 4 is particularly important because this is predominantly the sequence type that's associated with human cases of infection. But you can also see here that we can differentiate these sequence type 4 isolates very nicely by using pulse field gel electrophoresis, and we get some additional distinction here based on the pulsotypes. Sequence type 1 is also important because sequence type 1 tends to be the sequence type associated with colonization in the production environments. And again, PFGE type approaches will differentiate this to some extent. But what was rather intriguing, and I don't know really the answer to this, and since Ben is on the line, maybe we can ask him afterwards, the array showed us that we could distinguish between a lineage 1 type of sequence type 4, which was mainly associated with serotype 02, which one of my other graduate students, Niall Milan, had identified some years earlier, and what Ben called lineage 2 among sequence type 4, which was mainly composed of serotypes 03. And again, I'm not entirely sure if we ever came to a conclusion as to what distinguishes those from, I suppose, from an infection point of view. But as I say, maybe Ben will answer that question later on when we are finished with the presentation. So much so that again, when we go back to adaptation and we look at isolates of sequence type 1 from a powdered infant formula origin or a clinical origin, isolates of sequence type 4, either from a powdered infant formula origin or a clinical origin, we can see some different behaviors with respect to this swim assay under motility. And this gave us the notion that perhaps what we were observing here was a pathoadaptation, which was a phrase coined by Bentall at the time, which suggested to us that maybe these bacteria were doing different things in these environments, as I said, which was presumably underpinned genetically and being selected at the same time. So to some extent, we can see this, at least in a very early stage, from work done by Niall Milan in my group at the time, and then subsequently in collaboration with colleagues at CDC and the Malbrand Institute in Argentina, and also at Zurich. And for the first time, Niall was able to show that when we conducted an analysis of the colonization dynamics of Chronobacter species in a powdered infant formula production site, and that's what you're looking at the moment, a schematic drawing of that site, you could see very nicely that wherever you identified a Chronobacter isolate and then typed it using pulsed field gel electrophoresis, you got an invariable fingerprint, such as the one I show you here on the left-hand side of my slide. And every time we got such a fingerprint, you know, we just simply gave it a colored dot to illustrate the type of fingerprint we were looking at. And then we mapped all those colored dots back against the production site. And lo and behold, this is what we got. So we could see, for example, that we were getting a lot of colonization in or around parts of the production chain, which was the driest areas. But not alone that, it appears that some of these Chronobacter species disseminate through the production, possibly ending up in the final product, which is an absolute no-no. So this was one of those things, you know, a kind of a eureka moment the first time we saw it, and we thought, wow, this really gives us a picture of where colonization is happening with respect to Chronobacter, but it doesn't answer the question as to why. Needless to say, in collaboration then subsequently with our colleagues at CDC, and as I've mentioned, the Malbrand Institute and our colleagues in Zurich, we were able to develop a PulseNet protocol that was then part of that PulseNet methodological umbrella that was in use at the time. And so we started to use this over a much longer period of time, such that it would allow us to track the dynamics of what was happening in the production site. And this is a picture from, again, another one of my PhD students, Shane Cooney, who was looking at one of the 3 production sites that I mentioned at the introduction of my talk. And in this case, we were able to show that there was one particular pulsotype, which was associated with a bacterium that we called SP291, so that's it up here, which appeared to be consistently recovered from this particular powdered infant formula production site over a very long period of time. And that led us to ask the question, what was causing this particular adaptation to this particular site? If you looked at the tolerance to desiccation with time for this particular strain of Chronobacter sakazaki SP291, you can see here, this turquoise line here shows you that even after 32 months in storage, we could still recover this strain. So clearly, this was one of those organisms that had adapted to the production site. It was being selected by this low moisture condition, and presumably, as I said earlier on, this selection was being underpinned genetically. It was now a question of trying to understand, could we start to pick that mechanism apart? We could clearly show that SP291, if you like, was match fit, particularly in a powdered infant formula production environment, because it was able to withstand dry heat at 60 degrees for a period of at least 2 hours. And this was way better than the BAA type strain, as has been used in many studies. This was an outbreak strain that had been documented several years earlier in Tennessee. Similarly, a 12868 strain of unknown origin that had also been documented. And in comparison to those other important pathogens associated with powdered infant formula, Salmonella annatum and Salmonella ealing, which were associated with an outbreak in Britain several years earlier, the SP291 strain, as you can see here in my figure, is by far and away better adapted to life at 60-degree temperatures for a 2-hour period of time compared to any of these other strains. So what's the attribute that confers this phenomenon? And to do that, we decided to carry out an RNA-seq experiment to explore the transcriptome. I think everyone is probably very familiar with the use of whole genome sequencing, from the point of view of epidemiological analysis. And as I said, I'm not really going to say very much about it, but we were focused in on using a sequencing approach for RNA as opposed to DNA to try to get a picture of what was happening at the global level of gene expression. So that's exactly what we did. And in order to do that experiment, we had to go back to the genome of SP291 and completely close the genome. So we sequenced that strain to completion. It has a chromosome of about 4.3 megabases here, as you can see, about 4,000 coding sequences, and 3 residence plasmids ranging in size from about 4.4 thousand base pairs up to about 118,000 base pairs. So the design of the experiment is really a laboratory experiment, which is as close as we can make it to what we think is happening in the real world, in the production site. And the idea here was to initially take a look at the growth curve of SP291. So that's what you see here on the top right-hand side of my slide. And so this is a typical growth curve showing you the lag phase, the log phase. And we decided to take the culture at the early stationary phase, which we reckoned might be best suited to survival in the production environment. So the inset graph that you see here is a desiccation curve. And this desiccation curve is really important because what it's showing you here is that when you desiccate the early stationary phase culture, and then you follow recovery of that for a period of about 21 hours, you can see here that when you desiccate the culture, this is SP291 desiccated in liquid powdered infant formula, there's hardly any loss in bacterial numbers. Again, this kind of makes sense from what we think is happening in the production environment. Whereas if you compare it with survival in water, you see that the bacterium is not as adequately able to survive in water, nor is it adequately able to survive in TSB broth. So our experiment is a very simple one. We take SP291, grow to early stationary phase, as I say, recover total RNA there, desiccate part of that culture onto stainless steel coupons and recover RNA, and then sequence both sets, and then map these reads back to the genome so that we get a sort of a statistical profile of genes that are turned on and genes that are turned off. And then you do a lot more stats on it to try to identify changes that are highly upregulated, of which there were 361 genes signaling here, versus genes that are highly downregulated. And you can see from the pie chart at the bottom right, there are about 115 genes that were signaling here. Most of the bacterial genes here don't appear to change in terms of their gene expression at all. Again, we could confirm by qPCR versus the RNA-seq data that the genes that were upregulated by way of RNA-seq were similarly upregulated as detected by qPCR. So we knew that we had a pretty robust experiment in which we could start to draw some conclusions. So we started looking at genes that perhaps might give us a clue as to the adaptive phenomenon that we're trying to investigate here, which is survival under low moisture conditions. And when you start looking at genes associated with osmotic stress, there were about 37 of those genes that were signaling, and I've identified 16 of them here. And you can see the gene name and the functionality associated. And again, there's no surprises here in the context of the functionality of some of those genes. It all has to do with transporter mechanisms associated with osmoprotection of the bacterial cell as you withdraw moisture from its environment. So when you start looking at the mapping density in the transcriptomics experiment, you can see here very nicely that these genes are being switched on when the culture moves from an early stationary phase, and this is the sense strand of DNA, to a desiccated phase, where these 3 genes, BET-I, BET-A, and BET-B, get maximally expressed. And similarly, the positive regulator, BET-T, on the antisense strand is also upregulated during desiccation. Similarly, in the case of Pro-X, Pro-W, and Pro-U, which together make an ABC transporter that transports proline into the bacterial cell to again withstand the osmotic pressure, these genes are upregulated during desiccation and switched off during the early stationary phase growth. So this appears to consistently fit with what we expect the model to tell us. You can look more broadly then at the various cog categories within the bacterium to look at other factors that are signaling here. For example, in the case of amino acid metabolism, you can see the types and the nature of the pathways that are being affected here. Green meaning highly upregulated, pink meaning highly downregulated. And similarly, in the case of carbohydrate metabolism, and in fact, one to look at here is the very highly upregulated citric acid cycle, or the TCA cycle, if you want to call it that, and similarly, enzymatic pathways associated with glycolysis and gluconeogenesis. Also, in the energy metabolic pathways, oxidative phosphorylation appears to be highly upregulated. And again, that appears to be biochemically consistent with what I said with respect to the nature of the role played by many of these efflux pumps, which require energy in order to sustain their activity. So we could then start to think about a schematic to try to summarize all of this. And this is what we came up with. So this is a kind of a picture as to what we think is happening when you take Chronobacter sakazaki sp291 and you desiccate it. So you see a massive upregulation by way of the green colored arrows with respect to trehalose metabolism. So in particular, the genes OTSA, which converts glucose 6-phosphate into trehalose 6-phosphate, is highly upregulated. And similarly, the enzyme that clips off the phosphate group OTSB to convert trehalose 6-phosphate to trehalose is also highly upregulated. Here is the high upregulation of some of those enzymes associated with the citric acid cycle, and then the pentose phosphate pathway here also showing upregulation. So a lot of this has to do with the provision of energy sources and the provision of reducing power to keep the energetics of the cell stabilized so that these efflux pumps in the membrane can do their job. To see whether or not some of this is actually true, we knocked out the OTS-B and A genes that were responsible for that trehalose pathway that I showed you. And in order to show that there was no effect on the ability of the bacterium to grow, so this is sp291, an nalidixic acid mutant, versus sp291, the same nalidixic acid mutant with the OTS-A and B genes knocked out, the growth patterns were uncompromised. However, the desiccation curves looked very different. So if you like, in the wild type sp291 by way of this blue curve, there's that stabilization again in powdered infant formula that I alluded to once you desiccate the strain. Whereas when you knock out OTS A and B and you compromise the trehalose pathway, the survival is somewhat compromised as a result. The real question now is, do all of these Chronobacter sakazaki strains respond in exactly the same way to these low moisture conditions? Well, let's have a look. So we compared now what we saw in Chronobacter sakazaki sp291, which, as I showed you, is an environmental strain, versus a clinical comparator strain 29544. So we did exactly the same experiment again, grew 29544, the clinical strain, to early stationary phase, again harvested the culture at early stationary phase. And here you can see a comparative desiccation curve. So the green line here is the clinical strain 29544, and you can see that the numbers of cells drop off after about 4.5 hours, and you can't recover it after 4.5 hours. Whereas sp291 survives up to 4.5 hours and beyond. So we think there is an adaptive mechanism that is in play in sp291 that doesn't appear to be expressed to the same level of functionality in 29544, which may make that difference. And in fact, when we start to query the transcriptome of 29544, we start to see evidence of some of those very early slides that I alluded to by way of the potassium transporters. So here they are here. Some of these differentially expressed genes, here they are here, the osmoprotectants that I mentioned, and we could differentiate what we described as a primary response, so genes signaling very early on during the desiccation stress, versus genes that are signaling later during the desiccation stress, which we called a secondary response set of genes. Similarly, with respect to the differentially expressed gene transcriptional factors, we could see some differences when we compared 29544 versus sp291. And to try to summarize that then, if we took a look at some of the osmoprotectant mechanisms that we knew were in play in SP291, so for example, OPUCA, which encodes an ABC transporter, OTSB, which is the enzyme that clips off that phosphate group from trehalose 6-phosphate to produce trehalose, ProX, and betaine production, we could see here that these genes are highly expressed under desiccated conditions when you look at SP291, and much more so by comparison to the clinical strain 29544. So not all strains, it appears, behave in exactly the same way or respond in the same way to these imposed desiccated conditions. So to conclude then, what we can say about some of this work at least is that the molecular-based methods that we've been using here can support other conventional-based food safety control measures in order to try to add as much reassurance for producers as we can possibly give them. Understanding the microbial ecology of the production site is absolutely essential. And I really didn't speak enough about this, but I guess if I had more time, I could say a bit more about it. Differentiating and understanding the difference between these persistent isolates, such as SP291, for example, versus non-persistent strains, I think is very important, because studying their phenotypes may provide important clues as to how to improve food safety controls. And then, I suppose, as you might expect me to say, in terms of looking forward, the use of whole genome sequencing, where you're linking the genome to their phenotypes, is absolutely essential in terms of teasing apart how bacteria behave and adapt in these different hostile environments. And this will in time allow us to develop better and novel biomarkers to aid in their detection and to subsequently reduce risk to either the young infant, the neonate, or me, the older member of the community. And this was a labor of love done by lots of people over the years, located both at UCD, and this is some of our team at UCD. Carl Iverson, I mentioned, who is a real stalwart in this area. Niall Milan, who when he left my lab, went to work with Mead Johnson, as they were then, as their head of food safety in the US. Ben Tall and Gopal Gopinathro, who are on the call with us. So a huge amount of work by our colleagues at FDA, and also absolutely outstanding work by our colleagues at Zurich, Roger Stephan and Angelica Lerner. And with that, I thank you very much for your attention.
Thank you so much, Professor Fenny. Now let's get to the Q&A session. Feel free if you have questions. It's a rare opportunity for us to have an outstanding speaker like Professor Fenny. So go ahead, feel free. Well, we're waiting for, okay, here we go.
Oh, this is Gopal from CIPSA and FDA. Hi, Shay, nice to hear you again. Yeah, hi, and meet you virtually after a long time. I'm glad to see the overview of transcriptome and other related omic work. So one of my questions is, some of the genotypes Shabby and others have seen in your lab, how has anybody gone to look for the behavior of those genes in other strains that have been found in any other production plant?
Yeah.
Whether there is a consistent genetic lineage or conservation of genotypes that contribute to persistence in those environments.
Yeah, great question, Gopal. You always ask me tricky questions. So I think there are some overlaps here for sure. You know, I mean, I've really spent all of the talk literally talking about Chronobacter, but we could equally well have taken Salmonella. And in the case of Salmonella, we will see some overlapping features again, which are consistent with what I've shown you with respect to Chronobacter. So at least in the case of Salmonella and Chronobacter species, there appears to be some elements of commonality. I won't say absolutely the same, but let's say some elements of commonality when it comes to adapting to these osmotically stressed conditions. So again, like I said, with Chronobacter, we do see the role of transporters in the case of Salmonella, as I think you would expect anyway, but there are probably some features that are perhaps not very well defined by us, at least, that we haven't quite teased out yet that may distinguish Chronobacter and its behavior from Salmonella and its behavior in that same kind of environment, if that answers your question.
Yeah, it does. Thank you. Yeah, that's clarifying. Thanks.
Thank you. Thanks, Gopal.
And we have one, Magali, raise your hand. Go ahead.
Hi. Thank you, Dr. Fanning, for the presentation. It was very enlightening for me, so I can learn more about Chronobacter. I'm at the University of Maryland right now. I have two questions. One is when you describe these characteristics that help Chronobacter to survive in the environment, well, you mentioned several genes, some islands. I wanted to know if these features also have a relationship to possible pathogenicity. So because it may survive in the environment, but if it's not going to cause a disease, that's my first question.
Okay, that's a brilliant question. And thank you for asking, Magali, and thank you for your comments on the talk. It's a really important question because there's a little bit of confusion, at least in my mind, in the literature, because some people in the literature talk about, you know, when you have a dried strain in these conditions, it becomes more virulent. And I think this is what you're alluding to. And the question is, do we, okay, so maybe not. You can argue with me in a minute. But what I'm trying to get to in a rather roundabout way then is, we looked at this in Salmonella some time ago. And in the experiment that we did at that time, very similar to the ones I've described to you here, we see the spy islands switched off. So when Salmonella is desiccated, in the same way that I was describing the desiccation mechanism for Chronobacter, the spy islands appear to be switched off, at least in our experiments. So that would suggest to us that when the organism, and this is Salmonella now, is in a low moisture condition, it may not be as pathogenic as if it was in a planktonic state. We haven't done similar experiments in the case of Chronobacter, so I don't know how that would shake out.
Yeah, that was the direction I was going. I wanted to know if but one thing is what is happening with expression, but also it's later if those genes will be able to switch on when it becomes like it is in the food and it's going to be consumed. So
I suppose that's what I refer to then with that pathoadaptation term that I used earlier on, which is this ability of the bacterium to switch profiles, you know, depending on what the selective condition is at the time. And again, I think that's consistent with what we see. So, you know, if you think about it, when you dry down a bacterium, you know, you make it desiccated, you send it into a desiccated environment. One of the genes that you don't need expressed is flagella. It's not going to move. So the flagellar genes switch off. And this is exactly what we see. So then when you add moisture back into the condition, the flagellar genes get switched back on. But interestingly, and again, from work in Salmonella, one of the other interesting sets of genes that get switched on is phosphate metabolism. So if I can turn the tables on you and say, well, why do you think phosphate metabolism would get switched on when you go from a desiccated condition to an actively growing condition again? Why do you think that would happen?
It may require for, I don't know, energy. The energy
not so much energy.
No?
It actually requires it for nucleic acid synthesis. So DNA starts to get replicated, RNA starts to get made. So like, you know, like we really have begun to see lots of these things because we can sequence at depth and we can really look at the functionality of genes from that perspective, and we can ask those kinds of questions.
Oh, we also saw something similar with listeria in low temperature. When we grow it at low temperature, the flagella genes are turned off. So that's interesting. That's what we saw in my previous work at the University of Chile. Okay. My other question, it's a little bit off from this topic, but you were checking on the survival and environment, but also in the environment, there are other organisms that may be promoting their survival or not. So I want to learn to know if you have done some metagenomic studies about this. And that's my last question. Thank you very much.
You're very welcome. You must be reading my biography of some kind, but the answer to your question is yes. And if you look in foodborne pathogens and disease, I think it's back in July, there was a special issue which looked at next generation sequencing approaches. And there are some papers in that particular edition of the journal which describe using either 16S ribosomal RNA sequencing or shotgun metagenomics to describe, you know, the microbiota in an environment that you're talking about. And again, your question is very relevant here, because we can actually describe the microbiota in a powdered infant formula production site. So we know what the core microbiome looks like in terms of its taxonomy. What we would like to know, of course, is exactly the answer to your question, which is, can some of that core microbiome mitigate against the colonization? In other words, can you provide colonization resistance against Chronobacter? I don't know the answer to that question, but what I can tell you is that we did a very similar experiment to that several years earlier, where we looked at Listeria colonization of drains in a food production facility, and we saw exactly that. So we could see a completely different microbiome when Listeria was present versus when Listeria was absent. And then you could start to think about teasing apart and dissecting that microbiome to look for evidence of anti-Listerial activity, or in the case you're asking me, anti-Chronobacter or anti-Salmonella activity. And I think that's a worthwhile prospecting approach, if you like, because it's a natural approach to biological control.
Great. Bezar?
Hi, Seamus. It's great to have you back.
Hello.
I really enjoyed the presentation. It was fantastic. I have a couple of questions about, you mentioned you showed I think phylogeny of Chronobacter, and you showed 4 groups there. And among those ones, I think you mentioned 1 and 2 basically are more pathogenic, if or maybe have more prevalence versus group 3 and 4.
Yeah.
I was wondering if you did any comparative genomics to look at the differences between the two, if there is anything there.
It's a good question. No, we didn't, but maybe Gopal or Ben could comment on that, because they may have a better idea as to what the answer to that question might be. So let me see if they can answer it. Gopal, Ben?
Yeah, I shall take the first shot, and Ben is there. I haven't seen him activating his microphone, so I'll wait for I'll give some time for him to activate his microphone.
No,
so the microarray lineage, the way it separates is using nucleotide level differences in specific some parts of the gene which are well characterized or well read. So based on the allelic differences in those strains, in those regions we have looked at, the software, the program was able to identify slight differences among these 2 sets of genomes, the strains that are queried. So whether it has a direct genotypic and thus phenotypic implications, actually we have not looked at it. And as far as I know, we have not looked at it, and something that's worth exploring, because we have tons of data around that region from multiple strains, which were done after this analysis was done way back. So that's a good lead, and thank you for bringing it up again. I'm not sure Ben is online, unless he has.
No, I'm here. Can you hear me?
Yeah, yes, Ben, yeah.
Yeah, you know, we don't have a lot of information about that particular question. But let me just add this to the discussion. We do know that there are certain strains, say of sequence type 4, that are highly pathogenic in animal models, like the zebrafish model that Angelica Lerner uses in Roger Stefan's lab. But in that some of those same strains of sequence type 4, they are absolutely avirulent. So the question begs, why the difference? And to be honest with you, we haven't really looked at it.
Yeah, so I think Ben is referring to a very recent publications from our group where Shay is also an author. Some of the strains coming from him also in microorganisms, where those sequence type 4 strains originated from plants but display a difference in the virulence, even though they are whole genome level. They show a lot of similarities and cluster together. I think that part of the data is in the paper completely.
And I think Gopal, if I can just add to what you and Ben have just said, from the point of view of regulation, I think it's important to point this out. We don't make a distinction in terms of the species when we talk about regulation. So regulation looks at Chronobacter at the genus level, as opposed to trying to distinguish the different species, despite the fact of what we see in epidemiology. And actually, when the name change was done in 2005, 2006, it was done deliberately that way, such that until, as Ben and Gopal have pointed out, until such time as we knew more about these different species, we considered every species to be a pathogen.
Yeah, that's a very good point, actually. Yeah.
Do we have time? I have a follow-up.
It's up to Professor Fanning. He hasn't had her dinner yet, so
as you wish.
Okay, go ahead, please.
Seamus, you did basically high throughput sequencing. You looked at the genomes, and then you did the RNA-seq and look at the regulations of the genes and the changes that happen. My question is a bit general about the adaptation, right? Because certain genes have to exist in the genome so that they can activate it and basically show some adaptation. What is the role of transcription? I mean, when it comes to differences between different species with different degrees of pathogenicity or virulence, some of them do regulate these genes that help them adapt to these conditions, I think, better than others.
Yeah.
What is at the root of that? I mean, there are more genes that, we need to discover that basically affect the regulation of these genes.
Thank you. Thank you for that question. Again, another great question. And again, one that's very intriguing and difficult to answer. I mean, you're right. We don't have a complete picture here. And in fact, I didn't wish to give the impression that we had a complete picture either. This was just one aspect of, I suppose, an observation that from the point of view of the story, it makes sense in terms of what we're seeing. So, okay, it kind of might stack up, as the fellow says. But to try to answer your question, if I can answer it a slightly different way. Some time ago, we looked precisely at what you've just said, but in the context of antimicrobial resistance, and in particular at the regulation of efflux pumps at the membrane. So I mentioned efflux pumps in the case of transporters, that were transporting osmoprotectants. So there's about 5 different families of these, as you know. The expression of them is controlled either locally by, regulators that are within the genes themselves, or globally by other types of regulators that have a more overarching role. And it's interesting because if you take 2 different Salmonella enteritidis strains and you look at the expression and the regulation of a very prominent efflux pump called ACRAB, the regulation in a strain coming from a clinical environment could be very different to the regulation of a strain coming from a chicken. Do you follow me? So again, this is completely confusing as to, do we have an overarching mechanism here? Well, I suppose if you study type strains, the answer to that question is yes, you do, because that's the only strain you look at. But when you look at field strains, they behave differently. And I think all bets are off. And as Ben and Gopal said, when you have these different field strains in different environmental conditions, the regulatory network goes out the window. It is what it is, and you have to look at it within that context.
Thank you very much.
Welcome. Pleasure.
If I can ask one question, Shay, is it okay? Is it too late?
Yeah.
Okay,
no,
great.
It's not time for bed yet, Gopal.
Yeah, sure. I want to listen to your thoughts on this. I want to refer back to your response to Magali's question about the microbiome in production plants. there is always a temptation, like in an infant powder formula production plant, where it would help to do massive surveillance routinely. And to use for that, the temptation would be to go for culture-free, culture-independent detection methods, which is quicker, easier, and maybe economically effective too. But the microbiome-induced changes in the phenotypic expression of these pathogens may or may not be fully captured by the culture-independent testing, because the representative of the microbiome may be limited in a culture-independent testing in a routine basis. So what is your thought based on your SAFE project and other efforts you have done in the past for something like a sensitive infant formula production plant, culture-independent or culture-dependent routine swab-based enrichment method? Either which one of these would be more effective and useful in understanding any emerging pathogenic properties in those places where it has become more sensitive for the society because of the population involved?
What you've asked is a really critical question in terms of what is the future prospect for using culture independent approaches to looking at the dynamics of these different production environments. And I think it's fraught with problems. I mean, we've attempted to do it, and I wouldn't say, we've done it very well, but we've attempted to do it. And the more we think about using culture independent approaches for whatever application, thoughts come up in our head like, how much of the DNA that we sequence are we just going to throw away in the first dump because it's, plant DNA or it's bovine DNA and it's of no interest to us? And then we get down to the kind of couple of million reads, which are the phylogenetic profiles that we want. So, bacteria or archaea. And then we have to worry about, well, was the method we used to isolate DNA, was that compromising our ability to take all DNA, or are we sub-selecting? And then from the amplification protocols that we might use, again, are we being biased here? So there's a whole lot of stuff here, Gopal, that I think we still don't know the answer to. And so the routine application of culture independent approaches to study dynamics is really only, I think, at the starting point or close to the starting point. So to answer your question, I think for now, we need both approaches. We cannot just dump classical bacteriology that has stood the test of time. I keep saying this always. It has still stood the test of time. It will continue until such time as we have a better understanding of those vagaries associated with culture independent techniques. I don't think we can over and exchange one for the other, if that makes sense to you.
Of course. Thank you very much. This is exactly what I want to hear from you again and again, and convince myself not to go astray. Thank you.
Thanks, Gopal.
Yeah.
Any additional questions? I do have a couple of comments for Professor Fanning. I'm more like a Listeria and some Salmonella type of researcher. Sakizaki is relatively new to me. I was digging some literatures and I was trying to figure out knowledge gaps that is kind of existing in the Sakizaki research. I noticed a few things. One is I read an article or a publication by the First Steps Nutrition Trust. I don't know if you're aware of that. They published a summary about the bacterial contamination in powdered infant formula documents in August 2020. They collected some surveys regarding how prevalence of Sakizaki in commercial products, infant formula products. I was surprised because the data vary fairly significantly, and all the data looks pretty old to me. They are before like 2005 data. Not much recent survey was done. In particular, in the United States, there was only one publication in 2003, which identified a 22% positive rate for Sakizaki, and in other countries, somewhere varied from 1%, 5%, 70%, or so on and so forth. So I wonder why there's not much survey type of study is done to kind of look at current powder infant formula products and how exactly popular or kind of prevalent of certain types of strains or Sakazaki are present.
Yeah, maybe to try and address that question, Dr. Zhang, I would say that prior to 2005, any data that you are reading in the literature is circumspect to some extent because our isolation protocols weren't very good back then. I mean, it was really in or around the early 2000s, probably 2003 after, that we started to get media, you know, bacteriological media that was more dependable than the original FDA method that was available, which was based on, you know, bile agar. But I mean, that could detect pretty much anything. So that's the first thing I would say. The second thing I would say is that, again, recognizing the importance of the pathogen in terms of the production facility, you know, whilst powdered infant formula producers in some countries kind of really understood why it was important to detect this organism, not everybody did. And we see that reflected in issues that are happening in different countries. That's the second thing. And then I think the third thing is, you know, as we've begun to tease apart the epidemiology of the bacterium and we've begun to understand a little bit more about it, the question is to what was happening upstream of the production chain. So instead of powdered infant formula itself, what about the supply chains leading into it? And were there issues associated with those? And I can tell you there are very big issues associated with those because a lot of those protein producers, you know, who are basically milk companies or dairy companies, not powdered infant formula producers, have contamination issues. And those contamination issues could potentially get transmitted to a powdered infant formula manufacturer that potentially could compromise them. So really an appreciation of the importance of the organism and the potential impact it can have on a brand is relevant here. You have to sort of question the nature of the isolation protocols and the culturing protocols that people are using. And ultimately, you know, how relevant is this to your business? Because, you know, if you talk to Abbott, well, today they'll tell you that, you know, looking for this organism is their #1 priority. But maybe 6 months ago, it wasn't. And it's not just Abbott, it's everybody. You know, we take our eye off the ball.
Yeah, I agree. Second comment was from your slide, because you mentioned there's no reported study on infectious dose of Sakazaki. I understand that would be kind of a very challenging thing to do because the vulnerable population is pretty much premature babies. I was wondering if there is an animal model that is suitable for this type of.
Yeah, again, that's a good question. There is, when I made that slide a long time ago. So at that stage, it was still factually correct, but I suppose you're right to pick me up on it. It's perhaps not factually correct in terms of when you think about it in an animal model context. And so I think back in the kind of late 2000s, there were some papers that came from labs in the US that I just cannot remember off the top of my head at the minute, that did look at this and sort of estimated what they thought might be an infectious dose based on, I think, mice models or something like this, if I can vaguely remember it. But again, I don't recall that started gaining any amount of traction. What I think I would say is this, that, you know, the kind of individual you're dealing with, as I showed you in my slides, is incredibly vulnerable with virtually no immune system. So they can't mount really a serious immune challenge to the infection. So I would imagine that in that context, an infectious dose would be a very low number of viable bacteria.
I agree. One last question or maybe comment. When I was reading your AEM paper published and very recently, you mentioned a lot of the data today. You actually used a 4-hour desiccation process, and you provided 3 justifications why. We did similar study on Salmonella enteritis in peanut butter, and we kind of put inoculated Salmonella in peanut butter for 24 hours, 72 hours, and then we extract the RNA and we did a RNA sequencing study out of it. And as expected, less than 5% of the gene are actually being transcribed. So we didn't get much information out of the adaptation process. I was just wondering if you are going to do some similar things to look at the transcriptomes or maybe proteomes at a later stage of the desiccation adaptation in the future study?
Yeah, that would be a nice thing to do, but at the moment we don't have any plans to do anything like this. I mean, unfortunately, a lot of the research that we do is very much dependent on funds that we can get to support it. And maybe now is a good time to ask about Chronobacter again, given what's happened. But for the last number of years, it's been pretty barren in terms of funding.
I completely understand. Any additional questions from the audience? We are like 26 minutes out of our regular time. So if no, I would like to take this opportunity to thank Professor Fanning again for so much for your time, and also thank for all the audience who joined us for today's seminar.
So thank you very much, Dr. Zhang, and thank you for all the great questions. And I see comments coming in the chat. I'm very grateful that you liked the presentation, and it was a pleasure and an honor to do it. So thank you again for the invitation.
Okay. Okay, with that, have a very good evening.
Thank you.
Okay.
Thank you. And lovely to meet you all. Bye-bye.
Bye.
Bye-bye.
Okay.
Bye, Ben. Bye, Gopal.
Yep.